Good evening, students. Today we are going to learn about incomplete dominance. Now, till now we have learned that what is dominance. Mendel did work with the 7 contrasting characters, and luckily for all these 7 contrasting characters, he got the same results. What was the results? In F1 generation, he got 3 is to 1 phenotypic ratio. In F1 generation, 100%. And in F2 generation, he got 3 is to 1 phenotypic ratio and 1 is to 2 is to 1 genotypic ratio. So for all his 7 traits, he got this ratio and he gave the law of dominance. He said that for each factor, there are two alleles or two things. Only one can express itself in F1 generation. And the one which is expressing itself in F1 generation is dominant, and the other which is hidden, which is not able to express itself, is recessive. And in F2 generation, that is self cross of F1 generation, the result is 3 is to 1. So the hidden trait which was not seen in the F1 generation is seen in the F2 generation, and the ratio phenotypically was 1/4. That means 3 is to 1 ratio was there of phenotypic ratio. But with other, it was seen later on that with other traits, some traits were showing mixture of characteristics in F1 generation. That means if the true breeding dominant and true breeding recessive plants were crossed, the F1 generation was showing some blending, and it was showing a different character than the parents, and this was called as incomplete dominance. Now what is incomplete dominance means? As the term is suggesting, the dominance is not complete. That means the dominant gene is not completely dominating over the recessive gene. The dominant as well as recessive both are able to express themselves. That's why what we can see is the mixture of those two. So that is called incomplete dominance. So this is incomplete dominance. Let me give you this example. This is snapdragon flower, dog flower commonly called. These are red color flowers and these are white color flowers. So red color flowers are dominant and white are recessive. Now, if the red color flowers are crossed with white color flowers, the F1 generation is pink color flowers. So it is totally different from the parents. Both the parents, it is neither red nor white, it is pink. And this is not complying to the law of dominance of Mendel. So this is completely different phenomena. So it was found that, and who discovered this? The Correns discovered incomplete dominance, and this is also called partial dominance or semi-dominance. Means the dominant is not completely dominating. The recessive is also able to express itself. And when both are expressing itself, the result is incomplete dominance. Clear? So this is the inheritance in which allele for a specific character is not completely dominant over other allele. And this is called incomplete dominance. So the snapdragon flowers were crossed. The true breed red flower. True breed red flower means, true breed means homozygous. So true breed red flower means capital R, capital R. When they were crossed with white flower plants, they were white flower also true breeding. So homozygous, recessive, small r, small r. Now the F1 generation was pink. Mendel did not come across to such any phenomena because luckily what 7 traits he considered, no one showed this phenomena. So this was discovered afterwards, that if true breeding dominant organism is crossed with a true breeding recessive, the F1 generation is showing both the characteristics. That means in between, and it was pink, neither red nor pink. Now, when this pink was self-crossed, like we are doing for F2 generation in Mendelian crosses also. So this F2 generation showed 1:2:1 ratio. That means the ratio was genotypically same as the ratio of Mendelian cross. Because in Mendelian cross also, we saw the F2 generation is showing the ratio 1:2:1. And 1 is homozygous dominant, 2 is heterozygous, and 1 is homozygous recessive. So here the phenotypic as well as xenotypic ratio were same. So xenotypic ratio was same as Mendelian cross, but the phenotypic ratio was different from Mendelian cross. Mendelian cross phenotypic ratio was 3:1, whereas here in this cross, the phenotypic ratio is also 1:2:1. That means 25% are of red flowers, 50% were bearing pink flowers, and 25% were bearing white flowers. So what is the phenotypic ratio? 1:2:1. Whereas in case of Mendelian cross, it used to be 3:1. But here the ratio is different. Here the Mendelian ratio, genotypic ratio is same, but the phenotypic ratio is different from the Mendelian cross. Now, to understand this phenomena, first of all, we should understand what a gene is doing. Actually, gene कर क्या रहा है हमारी बॉडी में? What is gene? Gene is an information. एक information है जो इंफॉर्मेशन पास हो रही है पेरेंट से प्रोजेनी में. क्लियर? ओके. नाउ लेट्स टेक अ वेरी सिंपल एग्जांपल. सपोज देयर इज अ कोड, यू हैव मेड सम कोड, ताकि किसी और को पता ना चले. आपने एक कोड बनाया, आपका कोड है ABC. ठीक है, ABC कोड मतलब आप कहोगे कि अगर मैं कह रहा हूं ABC, तो इसका मतलब है कि आज बायोलॉजी की क्लास 5 बजे है. ठीक है. और अगर कोड है DEF, तो इसका मतलब है आज बायोलॉजी की क्लास 4 बजे है. दोनों कोड्स का डिफरेंट इंफॉर्मेशन है. ठीक है. अच्छा, अब क्या हुआ, दो ऐसे कोड्स है और एक ऐसा कोड है, ये दो ही कोड्स हैं, हमें पता है. अब इसके अलावा कोई आ गया एक और कोड LMN. अब LMN का कोई अपन ने इंफॉर्मेशन नहीं रखा है. पहले अपन ने क्लास में डिस्कस नहीं किया कि LMN का क्या इंफॉर्मेशन है, LMN कोड का. अब अगर वो कोड आ गया, सो दैट मीन्स दैट कोड हैज नो इंफॉर्मेशन. उसका कोई मतलब नहीं है, कोई उसका इंफॉर्मेशन नहीं है. क्लियर? अब जीन क्या कर रहा है? जीन भी यही कर रहा है. या तो जीन कोई इंफॉर्मेशन दे रहा है, नंबर 1, या तो जीन उस इंफॉर्मेशन को थोड़ा सा लेस एफिशिएंटली दे रहा है, मतलब पूरा नहीं दे रहा, या दूसरा थोड़ा सा मॉडिफाई करके दे रहा है, या तीसरा है जिसमें कोई इंफॉर्मेशन ही नहीं है, कोई एंजाइम नहीं. एक्चुअली जीन्स क्या कर रहे हैं? जीन्स आर मेकिंग प्रोटीन्स, और प्रोटीन्स फिर इंफॉर्मेशन दे रहे हैं, वो हम आगे क्लासेस में पढ़ेंगे. सो व्हाट विल हैपन एक्चुअली, this gene is, suppose this one gene is giving information for one enzyme. The one enzyme is produced by one gene. So one enzyme is produced by one code of gene. Another enzyme is produced by another code of gene. Now one code is such which is modifying the information for the production of enzyme. And one code is not producing any enzyme. तो होगा क्या? अगर मैंने आपको दो कोड दे दिए ABC and LMN, what would be the result? ABC is giving some information, but LMN is not giving any information. So what would be the result? The result would be that the biology class is at 5. Clear? Now suppose the codes are A, B, C and D, E, F. Then there are two informations. Now these two informations will express themselves. And what would be the result? The result would be some information taken from the first code and some from the other. That results in the incomplete dominance. That results that complete dominance is not. The gene is not being able to completely dominate. The gene is not exactly A, B, C, or the gene is not exactly C, D, E, F. The gene is something which is not completely giving the information, which is giving less efficient information. Or there can be gene which is not giving any information at all. For example, no information. So if no information and complete information is being crossed, the result is dominance of complete information. And if there is incomplete information and incomplete information is crossed, the result is some new information. If there is cross between the no information, the result would be no information at all. Clear? So this is by simple example I'm trying to tell you that what exactly the gene is doing. Now this red color flower is having two alleles. It is true breeding. True breeding means it is homozygous. Homozygous means it is capital R, capital R. Clear? Now this capital R is producing some enzyme, and this capital R is producing some enzyme. They are producing same type of enzymes, and those enzymes are capable of expressing red color. Clear? Now here, this white color flower is having homozygous. This is also true breeding. So this is true breeding white flag, true breeding recessive. Now true breeding recessive is having small r, small r. Now what happens normally, that this is expressing itself and this is not giving any enzyme. So what would be the result when capital R and small r are mixed? Always R is expressing it completely and small r is not expressing. So the result, the F1 generation is complete, showing the phenotypic character of R only, capital R only. So all are red, all are, would be red if it is complete dominance. But here it is a different case. Here there is different information of R. R is giving information for red color. But here the white is also giving some information. Now, when this white is also giving some information, the dominance of R is incomplete. And when the dominance of R is incomplete, there is production of pink color flowers. So pink is what? Incomplete dominance of R. Because pink is neither red nor white. So pink is a different color. So pink is showing that red color is not able to completely dominate itself. So this phenomena is called incomplete dominance. Now what happens when these pink colors, that is F1 generation, they are self-pollinated? What would be the result? Capital R, small r, these are the gametes of eggs and these are the gametes of sperm. And if we draw the Punnett square, we will get 25% red, capital R, capital R. 25% white, capital small r, small r. But 50% are pink. That is capital R, small r, capital R, small r. Why? Because this is 1:2:1 ratio genotypically as well as phenotypically. So in case of incomplete dominance, the phenotypic ratio and genotypic ratio remains same, which is not exactly what Mendelian ratio was. Mendelian ratio phenotypic was 3:1 and genotypic was 1:2:1. So here this is following Mendelian xenotypic ratio. So the phenotypic as well as xenotypic ratio both are same in case of incomplete dominance. So how can we define incomplete dominance? We can define that when the dominant gene is not able to express itself completely, the result in F1 generation is different character from the parent, and that is called incomplete dominance, because pink is neither red nor white. So if we see the cross, it is capital R are the xenotypes of parent, homozygous, and white they have taken W, this is W hybrid, just to show the hybrid they have taken. We are taken a small r and a small r. So R, capital R and small r are the gametes. So what would be the F1 generation? Capital R, small r. So just to show the hybrid, they have shown capital R, capital W, because here white is also expressing itself. So we cannot say that white is completely recessive. So it is written, not written in small letters, it is written in capital letters. So capital R, capital W. So here the gametes would be capital R, capital W. Now what would be the hybrid? Capital R, capital W. And this is pink in color, which is not resembling to either of the parents. And now if we self-pollinate this F1 generation, the result would be red, pink, pink and white. So the ratio is 1:2:1 phenotypic, and xenotypic also 1:2:1. So that means phenotypic and xenotypic ratio are same in incomplete dominance. So this is one phenomena where in F2 generation, the genotypic ratio remains same. Genotypic as well as phenotypic ratio remains same, which is not like the Mendelian cross. Because what Mendelian did, he got everywhere, he got in F1 generation 100% dominance, and in F2 generation 3:1.